That concludes my portion. I want to turn it over now to Kristen Don. Kristen Don is the Marine Reserves Program Leader, and she is going to now dive into Cascade Head Marine Reserve specifically and give you some details about some of the animals that live there and how they study this area and really what makes this site unique. So I'll turn it over to Kristen.
All right, thanks, Charlie. Everyone hear me okay? All right, awesome. Well, thank you so much. Really am excited to be part of this really cool event. So as Charlie mentioned, my name is Kristen. I work for the Oregon Department of Fish and Wildlife. I lead the Marine Reserves program. So our program's the lead on the management and scientific monitoring of Oregon's 5 marine reserve sites. So as Charlie mentioned, just wanted to share with you a little bit about the boundaries and the rules specifically for the Cascade Head site, and then we'll talk about what, from our knowledge and perspective, makes the Cascade Head Marine Reserve unique compared to other places on the coast or the other reserves. So next slide, please. All right, you saw this slide before. So Oregon's 5 sites, and next. And today, as Charlie mentioned, we're focused on the Cascade Head Marine Reserve here on the Central Coast. Next slide. Okay, so here is a map of the Cascade Head site. It is located just offshore from the town of Lincoln City. The Marine Reserve was, this site was established in 2014. The site stretches between Cascade Head, the headland at Cascade Head, and to the southern side of Lincoln City. Go forward, please, Charlie. So the Marine Reserve portion, so that's the portion that's close to all development and all fishing, is about 10 square miles. And then Charlie, if you hit again. And then there's 3 marine protected areas that surround the Marine Reserve, and those encompass another 23 square miles of ocean. Now, the marine protected area to the north does allow for the harvest of crab and salmon, so those are allowed, and it allows sport fishing for what are called ground fish species. So these are fish that are very frequently associated with rock habitat. So sport fishermen fishing out of a boat can still fish for those in the North Marine Protected Area. There's also a very skinny West Marine Protected Area, so just west of the reserve, and that allows also for salmon fishing and crab harvest, but no other type of fishing. And then the MPA to the south, it prohibits any use of net fishing gear, but otherwise all other legal fishing is allowed. The Salmon River flows into the North Marine Protected Area, so it's a source of freshwater input into the ocean. This is a very high, this area frequently experiences upwelling, which brings nutrient-rich water from the bottom up to the surface and makes for very productive food sources for other fish, invertebrates, and wildlife. So these are very rich waters. Charlie, if you wouldn't mind. Excellent. So this is a seafloor habitat map of just the Marine Reserve portion at Cascade Head. So what you see in dark gray is rock habitat on the seafloor. What you see in that sandy color is sand. In the lighter gray color is like mixed, so it's gravel mixed with sand typically. And then anything you see in green is land or an island. So you'll see the Marine Reserve here encompasses the northern portion of what's called the Siletz Reef. So the Siletz Reef goes about from the Salmon River all the way down past the Siletz Bay river mouth. So it's a really big reef. It's very highly diverse. It's got tons of different types of rock fishes, lingcod, kelp, tons of different types of invertebrate species. There is an island called Polly Rock that's also included in the northern portion of the reserve. That island is a part of the Oregon Islands National Wildlife Refuge, which is managed by the U.S. Fish and Wildlife Service. And then the Marine Reserve also includes, in the very northern section, Along the shoreline, it includes rocky intertidal habitat. And this reserve did experience pretty high fishing pressure predominantly from charter fishermen out of Depot Bay who'd come up here to fish for rockfish, greenlings, cabezon, lingcod, and then this is also a pretty popular area previously for commercial crabbing. But those have been closed since 2014, but those activities do still happen to the north, south, and west of the reserve. Let's see, what else did I want to mention about this site? So Lincoln City is the closest town. The closest port is Depot Bay. To the north is also Pacific City, which is an active fishing port. And on really good weather days, some of those fishing boats might come down to around this area previously. Boats from Newport also would sometimes, especially crabbers, venture into this area previously. So this was a very regularly fished area prior to closure. So next slide, please, Charlie. So I'm going to show you guys just a handful of photos that show us some underwater photos to get us an idea of what lies beneath the surface at Cascade Head. Most of us don't get to see for ourselves what is beneath the surface here in Oregon because the water is very cold. It's often dark and murky. But I did want to tell you, so we're doing research at these sites. We do intertidal research, but then in the subtidal waters, we do 4 types of surveys. So we do hook and line surveys that gets us really great information on fish. We do scuba surveys in shallow, rocky habitat areas. And then we have 2 video camera systems. And so a lot of the photos I'm going to show you are actually snapshots taken from these 2 video tools. So we have a video lander, which is a drop camera system that has 3 GoPro cameras attached to it. It just gets lower down, sits on the bottom of the sea floor. We let it sit there for 8 minutes. We roll it back up into the boat. We move the boat to the next sampling station, drop it back down to the sea floor. And then our other underwater visual tool is a remotely operated vehicle, also called an ROV. It has a high definition video camera attached to it. And we fly this just above the seafloor while it takes video, and we collect information on fish, invertebrates, habitats, and seaweeds with a remotely operated vehicle. So I'll give you guys a glimpse of what it looks like underwater in the reserve at Cascade Head. So next slide, Charlie. Okay, we're starting with an aerial view looking north at the Marine Reserve and the North Marine Protected Area. This aerial shot was actually taken from Oregon State Police's enforcement plane. They help us, they actually enforce the Marine Reserves for us, and our staff member Kelsey got to do a ride along with them and caught this really awesome photo on this beautiful day. So that's you see the Salmon River coming out into the North Marine Protected Area, and you see that beautiful headland at Cascade Head. Okay, next slide. This is that rocky intertidal area on the north portion of the Marine Reserve. Great place to go tide pooling. Next slide. Here's the ochre sea star, which we'll hear about later, as well as a lot of other invertebrates in the intertidal. Next slide. And then, of course, the elusive human species that we sometimes encounter at the reserve. Next slide. All right, so underwater. So this is a typical kind of better underwater visibility day. You can see it's still pretty dark, and you see this what's called marine snow. It's that white detritus in the water. That's really nutrient rich. But we have a basket star here. We have a lingcod. We have a couple rockfish and a sea cucumber. Next slide. This is a snapshot from the video taken with our video lander. We have this beautiful China rockfish with that big yellow stripe down it, and some other rockfishes in the background. Next slide. These orange fish are canary rockfish. They had been over harvested, but they did through some very aggressive fisheries management and some closed areas, this population did rebound and are now able to be fished again. We find lots of them at the Cascade Head Marine Reserve. We also see some perches and a sea anemone attached to one of the rocks in this photo. Next slide. This again was taken from our video lander. You see this cool China rockfish slipped right by us and swims through this little crevice right here. And on either side of the crevice are acorn barnacles. And in the video, you see the barnacles with their little hands out feeding, collecting plankton out of the water. It's really cool to see. Next slide. All right, again, these canary rockfish. We find them over rock habitat, but they also like to use sandy bottom habitat, or especially in those transitions between sand and rock habitat. Next slide, please. This is a screen grab from a remotely operated vehicle. We were flying along and came across this big boom of, these are sea cucumbers, these orange things laying on the ground, or with like the feathery things. Those are all sea cucumbers. We've also got some sea anemones laying around here, but it was about 25 yards of nothing but these sea cucumbers on this transect that the remotely operated vehicle was flying this one survey day. Next slide, please. Can anyone guess what these are? There's something on the sand here. Hundreds, thousands, probably tens of thousands of them. They're baby Dungeness crab. They're just littered the seafloor here, and they're scrambling around. In the video, you see them like scrambling around everywhere. It's really cool, but it's impressive how many there are when you come across them. Next slide, please. So that's just an underwater glimpse of the Cascade Head site. If you're interested more in the research that Oregon Department of Fish and Wildlife or any of our partners are doing, or if you want to see more video or underwater photos, or check out any of our reports or infographics, I'd encourage you to check out our website at OregonMarineReserves.com. And next slide, Charlie, final slide. And we also have a Flickr page, Oregon Marine Reserves Flickr page. These photos, we have tons of underwater photos here, also just research and action, also from outreach events. These are publicly available for anyone to use, so a great resource for folks for inspiration for art projects perhaps, but I'd encourage you to take a look at that too. And that's it from me.
All right, thank you so much, Kristen. It was great to hear from you. Again, I would encourage folks to go check out the website, OregonMarineReserves.com. I can't say enough good things about the video and some of the photography that's on that. A lot of their divers and individuals that get in the water have an eye for things, and they take beautiful, fantastic photos. And so next, we're gonna turn it over to Duncan Berry, who's on the community side of Cascade Head Marine Reserve, and an artist himself. Duncan has been involved since the conception of this marine reserves. He was involved with the community that helped in the process of identifying the area and designating it. And he's deeply involved in the broader UNESCO biosphere that makes up the surrounding area and encompasses the marine reserve as well. So we're going to turn it over to Duncan now to talk to us a little bit about the community.
Charlie, thank you. And also thanks for inviting me. I'm sitting within the Biosphere Reserve. In fact, the photo that you see, the middle meadow that's all green is my home. So we're coming live, streaming from near a marine reserve and in the Cascade Head Biosphere Reserve. What you see there is what Kristen was referring to as the Salmon River coming into its estuary and then meeting the Pacific at the head there. I thought I'd just give a quick snapshot from a community standpoint also who I am and how I'm involved in the community. What you see in this photograph too also is the West Wind property, which is in the lower left-hand corner, and the spit, some of you might be familiar with. and the Nature Conservancy and their headland, and then late successional reserve forests. So we have a very unique confluence of both land and sea connection. I was raised in the north coast of Oregon, so I grew up on the coast, and then I came when I was young to commercial fish. I think we're the only people that have ever commercially fished out of the Salmon River back in the 70s. And then my wife and I came back in 2006 to form the Western Stewardship Group, which is part of this patchwork of a conservation in this area, which is so unique. For the past 2 years, I've acted as a community-based organizer with Dan Twitchell of the Cascade Head Biosphere Reserve, which is a part of the UNESCO Biosphere Reserve Program. We're going to see some videos on that in a minute. But suffice to say that this is part of a network of over 700 biosphere reserves throughout the United States, or throughout the world. And this one is unique, and I think it's only one of 4 that throughout the world have conserved areas of the marine environment as well as the terrestrial environment. So it gives us a very, very unique convergence. Why don't we pop to the next slide, Charlie? So what you're looking at here is from Cascade Head looking down on our littoral spit. And the Biosphere Reserve basically terrestrially is about 17,000 acres. And what you're seeing to the right of that, under those approaching storm clouds, is the marine reserve itself. These are really rare and diverse ecosystems like Kristen was talking about. The Salmon River obviously bringing the estuarine complex being an incredible broadcaster of life and an incubator of life, both out into the ocean, but also upstream and into the air. We have densely forested uplands, a 2-mile-long basalt headland, and then these coastal prairies, which are really rare and very beautiful. Our mission within the biosphere, and this encapsulates the marine reserve, because 3 years ago in Dordogne, France, the UNESCO Biosphere Reserve Commission actually extended and recognized the work done by ODFW and the community teams, and extended the boundaries of the biosphere to include the marine reserve, which is a great move. Our mission, our sort of verb, which is really what I want to talk mostly about tonight, is twofold. One is to raise awareness of the incredible biological and cultural value of this place, and engage a wide range of citizens, scientists, and artists in the natural arts and sciences workshops and events that we hold. And that's why this event is really so perfect to me. I think there's a left brain and a right brain for us all in our relationship with the wild. And so the arts is an amazing to blend with and complement science. And so that's really been our way of involving, engaging the community. The other is just fostering rigorous scientific research and the conservation activities. We've been hearing a lot about that tonight. And to provide an inspiring example for managing wild and diverse ecosystems elsewhere. And one of the things I love about the Man and Biosphere program is that it designs humans into the landscape rather than it being just a conservation effort. One of the 3 legs of this tripod is development. So not in the sense of that we usually think of development, but how do humans take their place mindfully within an ecosystem and have sustainable livelihoods that are designed into and planned into a landscape. So just a quick snapshot. The next slide, Charlie, is we live in a, so I think you've seen this map. It's a little duplicative. This is the Biosphere Reserve, which incorporates some of the Salmon River watershed itself. And as you can see, with the Man and Biosphere program, it does take in Lincoln City as well. So next slide. In this time of COVID, I'd love to be able to tell you about our 2020 programs that are in full bloom. Unfortunately, we, like everyone else, are looking forward to going back to some of our activities, which are now been put in intermission. But this is an impact summary of the year of 2019, just a quick snapshot of the amount of people that we're engaging in the community. And from elsewhere, we've had people from all around the world. We had almost 900 folks involved in 13 different programs that we had last year. And these were a variety of different programs we're going to see some quick videos on. We also launched a major exhibit at the aquarium, which is still there, called Thanks Be to the Sea, which features panels and some information on the marine reserves and on the biosphere. And we basically focused on involving people either in the natural arts or in bioblitzes as ways to really tangibly have a hands-on experience in the field. So I'll beg your patience here. We're going to do 4 very quick videos here that show you a little bit more about what humans are doing within this landscape of the marine reserve and the biosphere. Our first one is just a general welcome, and I hope can everyone hear okay as well? Oh dear. Are you getting audio? Oh boy. Maybe we need to turn up the, maybe the volume? This might be just a visual rather than an audio visual presentation.
Give me one moment here. I'll see if it was maybe because I was on mute. I'll try and start this again.
Okay. There you go. Perfect.
Hi, my name is Duncan Berry, and I want to welcome you to the United Nations Biosphere Reserve at Cascade Head. This is one of the most beautiful, fertile places in the world. On the ground I'm standing on walk bear and cougar and elk. The skies above us are eagle and peregrine falcon and osprey. And in the rivers of the estuary that you see below us swim some of the most beautiful big salmon in the world. So biosphere reserves are part of the Man and Biosphere program of the United Nations. And they are a non-mandatory designation for places on Earth that are exquisitely and especially beautiful, and where man and nature have an opportunity to interact in a different way than we have done over the past several hundred years of industrial revolution. These biospheres are an experiment in how to balance economy with ecology. They are not purely conservation efforts, but rather a place where humans and the natural environment can interact in a different way. They're models for the future. There are over 600 of these biospheres worldwide, and in many other countries, they become magnets for ecotourism and one of the central forms of revenue for their economy. Here, they're lesser known with our national park systems, but we're lucky enough to have one right here on the central coast of Oregon. We hope you will come and join us and take part in the Cascade Head Biosphere Reserve.
So thank you, Charlie. And so I thought we'd just dive quickly into a couple more, one of which is our BioBlitz events. This is my co-organizer, Dan Twitchell.
Marine reserve that we have here, Cascade Head, is one of 5 on the coast. It is one of the living laboratories that is very important to our biosphere reserve. And there's a good deal of biology that's going on, spearheaded by the Oregon Department of Fish and Wildlife. So these living laboratories within the Biosphere Reserve are very important.
We got researchers out there working with volunteers.
The Marine Reserve activities that we like to present are these bioblitzes.
He's a male as well, right?
We go to a tide pool area off Rhodes End in Lincoln City. We have naturalists with us that can explain what people are seeing in tide pools.
It's about the size of a thumb.
We try to provide an opportunity for them to do that. And that's what these bioblitzes are about. You should treat the ocean as if your life depends on it, because it does. The ocean is paramount importance to everyone in the world. To the extent that we can really understand what's happening to it, we can perhaps take steps in time to, in fact, save it.
One of the things we did not talk about is the use of iNaturalist, which actually allows collection of data, even though it's citizen-based, collecting IDs over time of species in the marine reserve near shore environment. So we're going to turn to another way of engaging with our community here, and that is through our Gyotaku events. And I think that's our next slide. Right? Gyotaku being fish printing, and again, a way for people to enter into a relationship with these creatures.
Into these other lives, and for us to have a deeper relationship with these other species. You come to know something, and to know something that's this beautiful is to love it. And when you love it, then you'll be willing to step up and become involved with it and fight for it and protect it as it is precious.
There have been almost 600 people that we had in our Gyotaku workshops, and one of the hallmarks of it is when you take a piece of artwork home, you want stories to go with it. So while people are printing, we spend a lot of time talking about conservation and background on species, and it allows them about an hour and a half of education that they might not otherwise have sat still for. And then they have stories and they become ambassadors in their communities. So I think we have our message out to the public. And this is the exhibit I was mentioning to you earlier.
Welcome to Thanks Be to the Sea. This is our second exhibit. The first was at the Oregon Coast Aquarium, and today we're at the Lincoln City Cultural Center at the Chessman Gallery. It's printed directly on wood. We have over 40 panels here, and we're telling the story of interdependence of all humans and all life on Earth on the seas. With every beat of our heart, with every breath we take, with every thought we think. That's the real thing. One of the really special things about this exhibit that's never been shown before is the story of a gray whale that washed up at Westwind. This gray whale was a juvenile, 17 years old, who starved to death due to the fact that climate change has created a situation in the Gulf of Alaska where he couldn't get enough to eat to survive. And people ask me, so how did he die? I said, do you drive a car? Do you fly in an airplane? So if we think interdependence is a concept, we're very, very wrong, and this whale proves that. And then draw that line. This art form that we've utilized is about 150 years old and springs from a tradition that's often found in fish shops. It's not really a fine art thing. Gyo is fish, taku is rubbing. So we've utilized nature, or the fish here and the octopus, as the printing press. So you're actually literally brailing these beautiful creatures and having a relationship with them and creating art to witness them and to really honor them. To me, they're the superheroes in my life.
So I hope that gives you a snapshot of how humans are interrelating with both the Marine Reserve and the Biosphere Reserve at Cascade Head. I think our next slide wraps things up and gives you a link both to our site, but also to our scientist, Paul Robertson. So if you have interest in volunteering either on Marine Reserve activities or within the biosphere terrestrially, please reach out to us. And we had over 11 programs planned for spring and fall, many of them or a couple of them with the Lincoln County School District, all of which have been put on hold, and we're swinging around to spring next year. But please reach out to us, and we would love to engage with you, and we'll keep your name handy for when COVID releases its grip upon us. So thanks again, Charlie. Thanks for Surfrider and this program.
Thanks, Duncan. That was amazing. It's hard to walk away from that not inspired. So really amazing work. It's really great for me as kind of a science person too, to shift my brain and realize and remember why we all do this. And so we're gonna shift our brain now back to the science gears here a little bit. And hopefully you guys are inspired and impassioned now to learn about a critter a little bit. We're gonna dive into ochre stars, the who, what, and where of Oregon's ochre stars, which have been quite newsworthy over the past decade or so. And Silke has been researching these and was going to talk to us a little bit about Oregon's ochre stars. So I'm going to turn it over to Silke. Thank you very much for joining us.
Yeah, thank you, Charlie. Hi, everyone. Thanks so much for having me. As you mentioned, I am going to sort of 180 our train here a little bit and talk for a little while about my graduate school research, as well as the research of my lab group throughout Oregon, looking at the ochre stars that are so prominent and so iconic in our intertidal habitats here. Go ahead and advance to the next slide. But before I start talking about my science, I wanted to introduce myself a little bit to you all, and maybe share with you some of the ways in which my right brain also engages with the ocean. So I'm a PhD student at OSU. I'm actually entering my 5th year, so I've been here for a little while. And when I'm not doing science and working on my dissertation, I am an avid scuba diver, hiker, dog lover, skier, and traveler. I love to explore everything that Oregon has to offer. Next one. I grew up tide pooling in Washington and visiting the Oregon coast. I'm originally from the Pacific Northwest, and all the cool creatures that I saw there really sparked my interest and inspired me to study marine science. And I'm super proud to say that I am now, for a living, fulfilling the dreams of little 13-year-old me over there on the right by tide pooling for a living. So I kind of had that passion from a young age and have been lucky enough to follow it into graduate school. Next slide. And after I graduated from high school, I stuck out all of my challenging math and science classes and made a big move down to Southern California, where I did my undergraduate research at the University of California, Santa Barbara. This was not a bad place to study the ocean and to live. And I had the opportunity there to get involved with undergraduate research and do some of my first fieldwork and lab work and have some of those really fundamental research experiences. There, I studied the impacts of climate change on little tiny baby abalone, as well as juvenile sea urchins. And I learned a ton about climate science and realized how important it was that I work to understand how climate change will impact our oceans in the future. Next slide. Sorry, Charlie, could I get the next one? Oh, there we go. Thank you. And after 4 years in Santa Barbara, I knew that I really wanted to return to the Pacific Northwest. I absolutely love it up here. And so I applied to do my PhD at Oregon State with Professor Bruce Mengi in the Department of Integrative Biology. And I started in 2016, and my research has focused on understanding what determines how communities of organisms, a really fancy way for groups of species, respond to changes like climate change and disease outbreaks, which I'll talk more about in just a few minutes. In Oregon, I study how the rocky intertidal is responding to sea star wasting syndrome, which I will also talk more about in just a few minutes. And I'm also lucky enough to work in New Zealand, studying ocean acidification and how it affects both predators and their prey in the rocky intertidal. But enough about me. I'd like to introduce you to some of the members of my research group and talk a little bit about what we do. As I mentioned, I'm part of the Mengi-Lubchenco lab at OSU, and our research group is focused on community ecology in the rocky intertidal. And I know that community is a very familiar word to us all, but community ecology is a little bit more complicated. So I like to say that ecologists are the ultimate nosy neighbors. We really, really want to know who lives where and why. So as community ecologists who work in the intertidal, we study which animals live in different places, and we try to understand why they live where they do. A huge component of our research is centered around why sea stars are important in the intertidal and how they've been affected by a disease outbreak known as sea star wasting syndrome. I'm sure that this is very familiar to the majority of you who are listening, but I'll talk just a little bit about it just in case. Can we go to the next slide?
Sorry, there's a delay. So I'm advancing when you guys ask. It just takes a little while.
Oh, no worries. So it's just a picture of a bunch of very gory sea stars. But in 2013, researchers out on the rocks noticed that sea stars were melting and dying. And this was a very dramatic process that garnered a ton of attention, especially here on the Oregon coast, where people are so involved and active in our intertidal spaces. And this was a huge disease outbreak, and over 20 species overall were affected, ranging all the way from Alaska down to Baja. And this sea star wasting syndrome was actually the largest disease event that's been documented in a non-commercial species in the ocean to date. And the sea stars, sorry, next slide, whenever we get to it. When the sea stars get sick, they go through a really dramatic progression of symptoms. A lot of the media attention around this has dubbed this the zombie sea star disease, or the sea star zombie virus, and that's for a good reason. When these sea stars are affected, they start to lose their grip on the rocks, their bodies deflate and twist, they develop these lesions on top of their bodies, they lose arms, and then eventually they dissolve and die. And this may be something that you all have seen out on the rocks out tide pooling in the last few years. And this symptom process can happen really rapidly. Next slide, just whenever. And so these two pictures that are coming up are of the same sea star just 4 hours apart, transitioning from that losing grip stage all the way into that dissolving stage. So this is a really rapid and dramatic process. And next slide, and there's an animation, so if you just hit it a couple times, that would be totally fine. I threw this data in here mostly to just indicate that on the left side, red represents a decline, and to show you that between 75 and 99% of adult ochre stars died between 2013 and 2015. So for our really iconic ochre star in the intertidal, this was a devastating disease. Next one. So our group, our lab, works primarily in Oregon, but also does some work in California, and we have a number of collaborators along the West Coast, and we collect a ton of data about the sea stars. So some of the things we're monitoring right now include the percent of sea stars that are affected. So if we're seeing any sea star wasting out on the rocks, we record it. We record how severe any infected sea stars we may encounter might be. So are they dissolving? Are they twisted? Do they have some lesions? Kind of how are they doing? We also keep track of entire populations of sea stars at our research sites. So we measure their size and their density, how many of them we can find in a given area. And we also measure which species are affected. So primarily our ochre star, but we have seen other species in the intertidal be affected by this wasting disease as well. And what we've found since the initial outbreak in 2013 and 2014 is that the disease is still present at a low prevalence, especially in the summer. In the summer of 2019, we detected a smaller outbreak with about 40% of individuals affected at some of our field sites. And we're continuing to monitor this just with, again, the caveat that our fieldwork this summer has been relatively limited in the amount of time we've been able to spend out on the rocks and the amount of surveys we've been able to do. So, so far, so good. They seem to be doing okay. Next slide, please. And then I also want to share with you a little bit of this sort of why we care and why it matters that ochre stars have been so dramatically affected by this disease. And that's because they're incredibly important predators in the intertidal. As you can see from this photo on the left, ochre stars eat mussels, and they love to chow down on mussels in the intertidal. By doing this, they actually free up space for lots of species to live in the low intertidal zone. And in the absence of ochre stars, mussels often will take over that diverse low zone and crowd out lots of other species. And that's why we refer to them broadly as keystone species. So if you're familiar with sort of old stone architecture, you might have seen an arch with a keystone in the middle that holds it up. It's a similar concept in this food web where ochre stars are really important in maintaining the community structure. So again, that sort of who lives where and why. And in their absence, we see really dramatic changes in the species that are present. Next slide. And so these ochre stars are really key players in the intertidal. And in addition to collecting data just on the sea stars, we're looking at lots of other things. We're tracking population recovery and disease prevalence. We're investigating possible causes of the disease outbreak, which are still not super well understood. We are working on developing some contingency plans and improving our monitoring to respond better to marine disease outbreaks in the future. And then finally, we are tracking some of the ecological changes, like the mussel takeover that I mentioned, and how that's impacting our intertidal communities. So this photo on the right is one of my colleagues with one of our survey plots, which is near Port Orford, Oregon. This is not directly in the Redfish Rocks Marine Reserve, but it's quite close. And as you can see, there's a ton of mussels right in the middle of that plot. And we actually saw the mussel bed moved down 1.5 meters, which is pretty far in the absence of these sea stars. So these sea stars are really important ecologically. And then next slide. And outside of the broader work that our lab does, my research is focused very closely in on 2 other predators that, in the absence of this keystone predator, our ochre stars, might actually be able to make a big splash with their predation and eat enough mussels to affect that mussel takeover that I mentioned earlier. So I directly study this little predatory whelk over here on the left called Nucella austrina, or the northern striped dogwinkle, and another small sea star species called Leptosterius, or the 6-armed sea star, that wasn't as severely affected by sea star wasting. And I place them in the cages, like in the photo in the middle, to understand how their presence affects community structure. And so I'm asking whether these 2 predators can prevent the mussel takeover that we see when our ochre stars are removed or reduced in population as they were by sea star wasting. And I have, next slide, some pretty preliminary results to share with you that I'm nevertheless really excited by. At our site closest to the Marine Reserve, near Depot Bay, we found that there was actually no small predator effect on the community. So in some places, it seems that these predators aren't important in preventing that downward movement of the mussel bed. But at Cape Perpetua, the whelks are actually able to step in the absence of our ochre stars and eat enough mussels that they prevent that mussel takeover. And the same story is true at Cape Blanco, which is again near the Red Fish Rocks Marine Reserve, but for these little sea stars. So this is a really interesting story and is really facilitating a lot of better understanding of how our intertidal ecosystems can resist or be resilient to some of the big changes that we see in these ecosystems. So I was really excited to share that with you. That's a part of my dissertation research that's still ongoing and evolving. So this is a cool story. And in the last few minutes that I have, I wanted to quickly answer one of the most common questions that I get when people are seeing me out working in the intertidal or when I'm talking to people about what I do. And we're learning a ton from our research on sea star wasting syndrome about ecological impacts, and we're continuing to study how the ochre stars are doing. But I can use some of the data that we collect and some data from some of our collaborators, including people involved in and working with our Marine Reserve program, to give you a preliminary answer to this question. And so, sorry, next slide. There's a few things that matter a lot when we ask this question and when we as ecologists try to answer it. So in order for populations to recover, we need a few things to happen. One of those things is that we have to have lots of baby sea stars to grow up and take the place of the adults that we lost due to this disease. So that's down on the bottom left is the little blue sea star is a little baby ochre star, and it will grow up to be either orange or purple. And next slide. And luckily enough for us and for our beautiful ecosystems, we saw this. In 2013 and 2014, we had a surge of young sea stars that we detected in the Rocky Intertidal. And as you can see from the photo on the left of my colleague's hand, these are very, very small, but they grow very rapidly. And sorry, next slide. Not only do these stars need to exist as babies, they also need to survive and grow to the adult stage. Next slide. And so this is a type of figure that scientists use to track populations over time. And so from bottom to top, you can see that we go from small stars all the way up to large stars. And then we have time moving from left to right. And so that big red arrow represents sea star wasting. And you can see that we've lost a huge number of our medium to big size stars. But after sea star wasting, you can see in the blue circle, there's tons and tons and tons of little babies that we also call recruits that emerged after the wasting event happened. And if I can get one more slide forward, if you track the changes in sizes of the little dots over the last 4 years, you can see that those sea stars are growing and growing up to reach, they're getting closer to that sort of medium adult average sea star size. So we're really excited that we have these continuing sort of signs, promising signs of recovery. And then next slide. And then the last thing that we look at that tends to really matter is, again, that they're surviving to be adults, and then that the disease prevalence, so the amount of disease in the system, remains low, so that these sea stars that are growing can continue to survive, reproduce, make more baby sea stars, and keep on living in our intertidal habitats. And so next slide whenever you get a chance. And you can actually hit the next two, I think. And while we continue to see some signs of wasting, it's far less prevalent than when we initially detected it in 2013 and 2014. And when we do our surveys, see some large survivors and tons of juvenile ochre stars that don't appear to be wasting to the severity that they were in 2014. So really exciting and promising signs of recovery. We're cautiously optimistic for our sea stars in a lot of our field sites and in a lot of our rocky intertidal habitats. And with that, one more, thank you. I want to highlight the importance of our community scientists in the marine reserves in enabling us to be able to do this research. We mentioned iNaturalist, which is a wonderful tool. There's a couple of sea star health projects on there that I highly encourage you to check out. And if you're interested, we also have a community science reporting portal at seastarwasting.org. And then we also conduct and get support with surveys, outreach and education, and especially our marine reserve bioblitzes that facilitate our research and our work and are super helpful for us. And we hope that we can contribute to the marine reserve programs and these other programs in turn. So it's very much a collaborative effort, as is all the data that I've just shared with you. Next slide. And I'd finally like to acknowledge some of my coworkers and collaborators, as well as our research groups, the Partnership for Interdisciplinary Studies of Coastal Oceans, and the Marine Multi-Agency Rocky Intertidal Monitoring Network, or MARINE, and the Surfrider Foundation for having me. And then I have one question slide that I just want to throw up for a moment because it has our lab's website as well as our research group's website if you are interested in checking those out or interested in finding more information about what we do. And with that, I would be happy to take any questions that you have. Thank you so much.
Thanks so much, Silka. I'm gonna start my video again. I'm gonna ask our other panelists to unmute themselves at this point in time. And we are gonna go to a little Q&A period now. So we have at least one question here in the chat, and I will be kind of moderating those and directing them to our panelists. But if folks have questions, I encourage you to put them in the chat there, and I will call on one of our panelists to try and to answer those. So I'll start with this question from Gordon, which is, are ochre stars in captivity experiencing wasting disease? So, for example, the Oregon Coast Aquarium. I think that's a question for you, Silka.
Yeah, I'm happy to take this one. The short answer is yes, absolutely. We did see wasting syndrome in a number of our captive sea star populations. The slightly longer answer is that we have some sea stars that have been maintained in sterile, closed facilities. So the water that is in those tanks does not come into contact with the ocean. A lot of our local aquariums, like the Oregon Coast Aquarium or the Seattle Aquarium, pull water in from the ocean. And so we do have some populations of sea stars that we think haven't been exposed to the wasting and that haven't wasted, but we did see a ton of wasting in the majority of our aquarium facilities all over kind of the range of the disease.
I had a question just about that recruitment chart. So when I looked at that, what I saw was post sea star wasting, there was a lot of baby sea stars, like a lot more than you've ever seen in any of the other previous years. So what made them have more babies that year? You know what I'm saying? Like, is it because they were breaking up and making more sea stars? Like, is there a theory on that? Did everybody look around and see the other sea stars dying and say, hey, we better get busy? Like, what was that all about?
That's a great question. And the short answer is that there are a ton of unknowns in that sort of series of questions that you asked. This recruitment event that we saw post sea star wasting was actually one of the first times that scientists had documented those super, super tiny babies. Again, we call them recruits in the intertidal. And so we think there's a couple sort of different ideas floating around. One of the ideas is that they might have been hiding under the big sea stars, and so we just never really saw them. They were hard to find. And those little bubbles on the bottom of the chart, I want to emphasize that those have an arm length of about 2 to 3 to 4 to 5 millimeters. So they're like smaller. I find sea stars in the intertidal regularly that are like smaller than my pinky fingernail. They're very, very small. So we think that there is a potential that, yeah, the adults all spawned, they all died, and then there was lots of sort of space for them, the little babies, to come back in and be really successful in the intertidal because they weren't competing for prey or for space with the adults and potentially with their parents. So but there's still a ton of unknowns in that, but this was sort of an unprecedented opportunity to observe this big, we refer to them often as recruitment events of this species for the first time.
Sorry, I muted myself. This other question kind of feeds into that. And Soka, I think you'd be best to answer this. How do sea stars reproduce? And my understanding is both sexually and asexually. So maybe you want to describe both of those processes?
Yeah, that's correct. So sea stars can reproduce asexually, so that means without sex. And that's by splitting. So if a sea star is damaged, it has this incredible ability to regrow its limbs. And if a sea star is, say, split in half, it will actually regrow 2 more sea stars as a result. And that's one of the ways that they can reproduce. The other method of reproduction that they have is sexual reproduction. So if you've ever been out in the intertidal and seen a sea star that looks like it's leaking orange goo into a tide pool, they're actually releasing eggs. So the female sea stars release eggs, males release sperm. They mix in the water column, and they actually go through a number of extremely beautiful and really incredible larval phases out in the open ocean as part of the plankton. So the collection of tiny drifting organisms that hangs out in the open ocean water column. And then when they're ready, they actually come back to the intertidal, undergo metamorphosis, very similar to the way that a caterpillar turns into a butterfly, except instead of a caterpillar, it's this really wild kind of hula dancing looking little larva, and then it turns into a very, very tiny sea star. So that's the other method by which these sea stars are able to reproduce.
A lot of art in that.
Yes, yeah. I can put a link to an image of one of the larvae in the chat really quick. I think they're just like amazingly cool.
Beautiful. Here's another question related to sea star wasting, since we're on that, which is what caused it?
That's a great question, and it's another answer to which I can maybe give you more unknowns than knowns at this time. So we actually haven't identified one thing that's caused sea star wasting. We don't have a candidate virus or bacteria. We don't know is kind of the short answer to that question. There were a number of things. I actually have a slide that's like, why is it hard to study diseases in sea stars? There were a number of things that made it really challenging for us to understand the spread of the outbreak. And we also have relatively limited information about the immune systems and the potential immune response of sea stars. So there are a number of things that were really challenging for, there's actually a subcategory of marine epidemiologists, which is a group that I would not personally consider myself to be a part of, but they've been working really hard on trying to sort of figure this out, and we don't have a definitive answer yet.
So how will the delay or cancellation in research caused by COVID impact sort of long-term monitoring at these sites? I think that's a question for Kristen.
Sure, and then maybe Silky can talk about for the OSU and Maria. Well, it's always a bummer to have a year that doesn't have any data collected. However, scientists are very used to this happening. Sometimes your equipment breaks or something happens and you lose data that way. Sometimes you run out of funding for a year or two. Sometimes a worldwide pandemic hits and you can't safely go out in the field. The part of it is collecting enough long time series of data can still help fill in some of those gaps, or able to still see trends over time if you have a long enough data series. But there's also some ways using some statistical models that you can kind of fill in some of those data as well. And we've been exploring that, especially for some of our hook and line data, whether we might be able to do some math to help fill in some of those years. So it's not ideal, but there are ways to deal with those gaps. And Silky, I don't know about, I'm sure you guys have been talking about this at your lab, and you guys have some very long data series, which is amazing.
Yeah, we've been lucky enough to be able to work in a very limited capacity. We submitted a plan for resumption of research to Oregon State, and they were okay with us sort of doing these very stealthy kind of one car, one person trips out to the field, where we don't make any stops and are able to remain socially distanced and outdoors while we're working. So we've been able to do kind of a few of our most important long-term data collection things. But yeah, we've just had to kind of mothball a bunch of our projects with the understanding that, yeah, sometimes you miss a year, sometimes it's not possible to sample, and we'll resume them next summer.
All right. Well, that, guys, I think concludes our science and background education on the Marine Reserve portion of our event. I'm really excited now for us to make some art. So we're going to be turning it over here to John in just a second. I want to walk through a few logistics that hopefully will help us all. One, thank you all for participating. I'd like to thank again our panelists for taking time out of the evening and sharing their story and sharing their information. We will follow up with an email with more information and links to some of the stuff that was shared here, so that you'll be able to explore some of these things on your own. As we move into the art class portion, if you would like to, you may now turn on your video, but please keep yourself all on mute. We're gonna have you all automatically muted. So, for now, keep yourself on mute, but if you'd like to go ahead and turn on your video, you can do that. We think that will help with the art portion. So if you have a question, you can raise your hand. That's the easiest way to sort of get my attention so that I can grab John or somebody else. Where is that? If you click on the participants, you'll see in the bottom sort of corner right there, there's a little raise hand icon. And you can press that. We'll see that and flag that, and then we can kind of call on you and unmute you, so that people are not all talking over top of each other, and that way we can call on individuals. To keep the video of John large, I'm going to stop screen sharing here at the end of this, and for you to get his screen large so that you can see him full screen, you're going to need to pin his video. How do you do that? Well, if you look at this little slide, it kind of explains. If you scroll your mouse over top of John, wave at everybody so they know who you are. That's John. All right. So when you mouse over top of John right there, after I stop my screen share, you're going to mouse over top of him. You click on that little 3-dot icon and you can click pin video, and that will make John full screen for you so you can see what's going on. All right. We will be following up with more information on this, but as John mentioned early on, the whole goal here is collaboration. You are all part of a larger art piece, a mobile that is going to be put together by Elizabeth Jones Art Center and John. We encourage you to mail your pieces in. 516 Northwest 14th Ave, Portland, Oregon. We will be emailing you out this information as well as providing you with any, like if there's any questions, you can follow up with us, or if there's, if you guys want, in certain areas, we might be planning some drops with different businesses if that makes it easier and we can mail it in. So we'll see, we'll be following up with you more on that. The goal tonight though is just to make some really cool art and hopefully at the end be able to get this to John for a really neat collaborative project. So with that, I'm going to stop my screen share. Remember to pin John to the top of your video. And John, we're going to let you take it away.
Thank you very much. Well, welcome everybody. Let me just give you a little background about the exhibit that we'll be doing in October. It is a fundraiser for Surfrider here in Portland, where the gallery is. We are in the process of creating a 2-foot by 1 mile mural that will include every mile of the California, Oregon, and Washington coastline. So every mile will have a painting that'll be 2 foot by 4 foot, and we put them all together in geographic order, starting at the Mexican California border at Tijuana, San Diego, and going all the way up to Washington. So we will be having an exhibit of paintings, and the paintings that we will have will all be a fundraiser for Surfrider Portland. Now we have this really beautiful space. In fact, I'm going to show it to you. See, there's the gallery. See how nice and big that is? Now, what we're going to do is have a mobile that is 25 foot in diameter and 10 feet wide. And here's what it will look like. Let me see if I can bring this up. How well can you guys see that? Well, there it is. And that is just a section of it. Now, what we will be doing tonight will be this little guy right here. The painted sea anemone, or the painted anemone. Now, there was some difficulty with getting all the materials and all the information from me to Bree on time. So what I'm going to do is bring up on my screen. Could you help me with this, Josh? Let me get my mask on. Okay, Josh is going to bring it up on your screen so that if you are unable to download it.
Attendees need to be able to.
Pardon?
The host unblock the screen share.
So what's happening, Josh?
If the host could enable screen sharing. Cool, perfect, thank you. There you go.
Okay. Okay, so I'm trying to make it.
See that?
Yeah, can you make it a little bigger? A little bit smaller right there, a little bit more, a little bit less, a little smaller. Just a little small. Okay. Yeah. Okay, so if you were unable to get the template for that, there should be one on your screen right now, and you can put a piece of paper over on your computer, and you can outline that. So this is what we are looking to do. So once we have the template, is there anybody that needs this up on the screen, or should I move on? Okay, so the first step is, so we have toothpicks. This is really hard, so have patience with me because I haven't taught a class on Zoom yet, and I'm finding it difficult to get all the things right. So what you're going to do is to create a jig, and we're doing the jig with the cardboard. So you have 4 pieces of cardboard, and then we're putting toothpicks into it. And it's kind of hard, and then if you have some pliers.
John, if you can hold that a little bit higher, the bottom of it is cut off.
Okay,
there
good,
you go.
Good. So when I have these flat on a table, I can push them through with both of my hands. And it's difficult with this, so I need to use pliers. But
So describing this just to make sure if anyone is having trouble seeing on the video, John has his stack of cardboard with the paper on top of it, and
yes.
then is sticking the toothpicks along the outline on the piece of paper.
And the main areas that you want to get, see, once we have this jig, then you're going to take the wire, and then we're going to run the wire along the toothpicks, and then with that we will have this. If you can see the black.
So it looks like, John, it's important to get the toothpicks anywhere there will be a bend in the wire.
Yeah.
It's really important.
Yes.
And then how many did you put along the line in the bottom? Is it just in the corners on the bottom?
Yeah, just in the corners.
Okay. So wherever there's a change in the shape, there should be a toothpick.
So just take your time when you're doing this. Now this is really difficult for me because I'm trying to do it, holding it up to the camera. But hopefully you're doing it on a table.
Yes, the links were emailed out earlier today, and I am going to drop the link in the chat as well.
So coming up, we have all kinds of like beautiful creatures that we're going to be making for the mobile.
The link is in the chat, and the PDF file that it is linked to, the anemone is the first one in that PDF file.
Now I can't tell how many students I have. Could somebody tell me how many people are involved right now?
Yes, you have 18 students right now.
Looks like some of them have multiples, though. You may have more than that.
Yes, we have 18 connections, but there are multiple people on quite a few of them.
Well, as I told Bree, I think this first start is maybe a little rocky. You know, just that I'm on a learning curve, and yeah, actually pliers are a good way to do it too, where you just kind of like shove them in there. But so anybody who would like to work this Saturday or Sunday, go to our website, elizabethjones.art. And on Saturday and Sunday at 5 o'clock, I will be doing another class. And we'll look at some of the other creatures, or we'll just finish, you know, we'll look at how this one has turned out.
So John, one of your students put in the chat that she's using a push pin to pre-make the holes, and then her toothpicks are going in a lot easier.
Oh, sounds like a great idea. Sounds like a really good idea.
So John, for the class this weekend, will that be streaming directly on your website, or there will just be links on your website?
Yes, it'll be a Zoom class, and
okay,
and there will be a link on our website.
Great, thank you.
And
we'll be sure to include the link in our follow-up email to everyone.
Yeah. You know, what I tried to do is see, like now normally, if we would do this, the jig would involve you know, a hammer and nails and wood, but I was trying to think of materials that that you have right at the house, you know, and that you wouldn't need anything, you know, special like a hammer and nails and the other thing is I worked at getting materials that you could get at Ace Hardware, you know, just because a lot of the locations you don't have an art store. So I wanted to, you know, so that's why I tried to have everything that you can get at your local stores. You know, I have to admit, this is a little odd teaching. I've taught before, but it's like, what's strange is not having people going, "Hey, John," you know, or something like that. Oh, wait, there I am. Oh. Okay, so this
John, about how many do you have in yours?
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 25, and just everywhere where it changes direction, I put a toothpick. Now, is there a way that I could, can I take a tally for those that are ready for the next step?
I see a thumbs up. How about, oh, yep.
Not yet.
Okay, good.
We need a little time.
Yeah, let's give folks maybe let's give folks till 5 after to sort of finish up. So just a few more minutes.
So, so in regard to some of the materials, so this is what the brass rods will be used for. You know, these for, you know, what we put the fishes on and the different creatures, you know, for the balance.
It looks like you have the brass shims for some of your creatures and then the paint color swatches on some of the other creatures.
Yeah, yeah. Well, Bree and I were talking about a list of the creatures that could work in the you know, for the mobile. So these guys here, why these are the juvenile fish, and as Bree put in quotations, not sure how to represent those, but it's a particularly good site for juveniles of several species. So that's what those guys are. And those are a bit easier to make.
So how many creatures in total, John, are there for this marine reserve? 4?
Oh, I got 5. I got 5 creatures. I've got the, you know, there's our ochre sea star that we were just talking about. And then, let's see, you can see it.
Oh yeah.
And then up here is the lingcod.
I like that lingcod.
This is the cabezon. Can you see that? Okay. There we go.
Oh, cool.
And then over here, this is the, there we go. And here we have the canary rockfish. What's really nice about these, as this moves, it feels kind of like it is in the ocean, you know, because there's that, you know, kind of that rhythm as it moves. So, yeah, this is what happens often. You start moving things and they get tangled. There. There we go. How is everybody doing?
So for the people that are still working on the jig, I can see a couple are still in deep concentration with their toothpicks. So maybe just a couple more minutes.
Okay, good, good.
Yeah, some people are very fast with their toothpicks. Oh, one student said their artwork just got the melting disease and they'll have to try again.
Oh no.
It's abstract.
Not the anemones too. Well, those are all wonderful presentations. I love the ocean too, and it's always amazing to see all the animals, how they live, you know, in underneath our sites, you know, beyond our site.
Yes, those were really great presentations, and the underwater videos and all of the intertidal photos, and hearing from Duncan also about the community support. And, you know, Silka also mentioned, I know ODFW has community scientists as well that are helping to collect data. So much like this mobile that we're all making together, that's also how a lot of the scientific information is coming together for monitoring these marine reserves, which is a really nice connection.
So Bree, have you done classes or done anything like this online?
Not art classes. We've hosted presentations, but we haven't had anything where we were making something together. I think the closest thing we've done is trivia, maybe, where we have a lot of different people interacting, but nothing like this. We don't do a lot of art, and that's one of the reasons we were so excited about this.
Oh, great. Well, I'm trying to think how this could be done better, and it seems like I need a camera that's just on the artwork so that I can so that I can work on a table, you know, rather than like holding it up and trying to. So that's something that I'll need to look into.
Yeah, and we will be sending out as part of our follow-up email. As we mentioned, this is the first in a series of 5 events, so we're really looking forward to getting feedback from you all about the presentations and the art class as well. So we're learning, and hopefully we can improve for each next event.
Yeah. I hope I do improve.
Okay, is everyone ready for the next step? Thumbs up so far. Okay.
Are we all
It
good?
looks like we're ready.
Okay, so then take your wire that you have. Now you, of course, will be doing it on your table. And I'm trying to, okay, so. Then you take your jig and you go over and under. Oops, and I got one that broke.
Don, can you just point your camera down a little more? Or yeah, or hold it up. There you go.
See if I can do it this way. Ah, I'm breaking all my toothpicks, darn. There.
So are you weaving in and out?
Yes, I am.
Okay.
Here, hold on.
So if you start on the outside of one toothpick, then you'll go on the inside of the next one, and then the outside.
Now something, you can use your scissors for cutting the wire. And I'm going to start over because in the process of doing that, I've broken different things. Oops. Okay. It's like Bree was saying, it's over and under. You can use your scissors to like push that down like that. A little stronger connection.
John, we have a question about how you get the wire to lay flat.
You just have to kind of force it. You can, like if it's not laying totally flat, you can use your scissors to like smash it down. But what's going to happen what's going to happen is once we get this threaded through this, we'll take the toothpicks off, and then we will look at the pattern of the anemone, and then we'll like bend it to that pattern. So this is the first step, and then there's a second step to making it fit right. I have to admit, I'm watching myself do this, and it's, I don't know if I'd have much confidence if I was watching me, but there we go.
I imagine it's very difficult standing and not having the tabletop.
Yeah. The other thing you could do, if it's not, if lying flat is problematic, you know, it's like really coming up or something, you can put a little piece of tape on it. And that's all right. But just take your time and see, mine's not lying flat over here, but I'm not going to worry about that for right now. I'm going to, and you just go through, and then at the, when we get this whole thing threaded, we'll be able to correct things like that. I want to thank everybody that's out here doing this. You're going to be part of a very amazing piece of art, and I think once it's finished, you'll be very proud to have been part of it.
So John, are you going around the shape just once, or are you going around multiple times?
Just once.
Okay.
And just weaving in and out. Let's see. So, and then here I am at the beginning again. I'm just going to twist it until everybody is ready. And this is what you could do. You could just twist it like that, and that way it'll hold its place while we wait for everybody to finish.
So once you get back to the beginning, twist it like a twist tie.
Yeah, and then here, excuse me a second. Excuse me a second. So what I have here is a screwdriver, and I'm going to use that to try to, you know, this area right here is all kind of bunched up, and it'll help me like separate it a little bit, like that, like that.
So after you twisted it off, now you're just working a little more on the shape.
Yeah, yeah. they're beautiful creatures, and I really agree with. Now, who was it that talked about the United Nations reserves? I'm sorry. Duncan, yeah, you know how he was talking about how important. Well, I'll put it in my words. I'm not sure if these were his words, but things like beauty is important because it's because when we find things beautiful, that's the first step towards loving them, and that's when we start caring enough about things to protect them, and to work at that. It's just how we do things with the way that we love things, and beauty and art and all that can help us realize that kind of thing. So I'm trying to make this a little better here. And this is a lot easier to do if I was, had it flat. But then once I get this off of here, then we can like twist it around and flatten it. Just bend the wire. The odd thing about like a Zoom class is because I can't see the students and I don't know what's going on.
Oh, Stephanie is showing you her work. Looking good. And I see everyone heads down, focused.
Good. And I want to give a special thanks to Bree, because as we've developed this art part, I was not quite sure how it was going to work. How are we going to make a mobile? Because I didn't want to have a parts list that that you could only get at like Blick's art store in downtown Portland, because of course that doesn't make much sense for what we're doing. And anyways, Bree's been very patient, and she's had a lot of really good advice, and I want to thank her for that.
You're very welcome, John. Thank you for leading this art class.
Oh, sure. Now, see, at this point, I'm looking at my painted anemone, and I'm just thinking, that looks really awesome. What do you guys think?
You got some thumbs up.
Thumbs up? Okay, good. But there's the, see, what's really interesting about this kind of mobile is the wire becomes like a drawing, like you think of a line drawing, and this could be a line drawing of a painted anemone, and yeah, there we go.
John, just a time check. We're at 7:20.
Okay.
And we're slated to go until 7:30, but we can keep the Zoom going if people need to wrap up the.
Well, why don't we plan till 7:45? How does that sound to everybody? Are we getting thumbs
Yes.
up?
We're getting thumbs up and nodding.
Okay. And then should we go for another till 7:25 on this, and then we move on?
Yes, it looks like people are still working on their wire wrapping. Is that correct? Some people are finished.
Okay, well, let's just wait for everybody to finish, and then we'll move on. And if we don't quite get everything done now, then well, we'll just figure that out. Trying to get where I've like bent the wire a little too much. And look at that. That was Inga Hazen. She's our director of exhibitions. Normally she would give me a heads up so I could get my mask on, but guess not. So if you,
Okay, can you just let us know in the chat if you still need a couple of minutes? Nathan, right now I'm seeing people who are ready for the next step.
should we go for a next step?
It looks like we're ready, yeah.
Okay, so so next we're gonna clip the wires. So you can use scissors if you want. That works. But of course, it's better if you have wire clippers. So that's best. Okay, so now what we're doing is we're gonna lift this off like that.
And John, did you leave a little bit of wire or did you clip it close to where you twisted it?
Well, we're still gonna twist it, and
okay.
So it still needs to be twisted.
There's a little bit of a tail there.
Yeah. So then I'm gonna, after all that work of putting the toothpicks in, I'm taking them out.
It looks like Stephanie got hers out without removing the toothpicks. Split it off. Great. Oh, that's awesome.
So like for this particular animal, it doesn't really matter, but for some of the, like for the canary rockfish, like this fish here, we will need to like look at the pattern and try to form it, to the drawing part of it. But for this guy, it's pretty easy. See now, that's not perfect, but it looks kind of nice, I think. So it doesn't have to be perfect. And we can like flatten it out.
Yeah, those anemones all look different depending on if they're out of the water, in the water, how open they are.
Then I just twisted that, and I'm gonna like make it part of the bottom part.
Okay, so you twisted the wires together and now you're twisting it around that bottom piece.
And then just, you know, this is the model, and, you know, in many ways that doesn't look much like the model, but I think it works anyways. What do you think, Bree? Does it work?
Yes.
Yeah. Okay, and then take this, where you have that sticking out, you can clip it like that. And there's that part. Okay, so for the next part, you get your brass.
And if people have the paint color swatches instead of the brass, that was the alternative back for this.
The pink colored.
Yeah.
Okay, so okay, I'm sorry. So there's the brass, and then what else is there that people have?
Oh, I was saying some folks might have the paint color swatches.
Oh, the color swatches. Yes. Let me show you the brass, and then I will show you the color swatches. So for the brass, take a pen. And then just trace it like this.
Okay, and so on that PDF, there's a smaller anemone up in the top right corner, and that is what John is tracing onto the brass shim with is that a regular ballpoint pen?
That's a regular ballpoint pen.
Looks like you're going around a few times.
Okay. So what that does is it creates an outline. Can you see that?
Yeah.
Okay. So trying to find my scissors. Hold on a second. So with the scissors, just cut that out very carefully. And etc. Okay, so those who have the brass, I will let you continue with that, and then I will show you what the people with the color swatches.
John, the pack of shims has different thicknesses. Does it matter which thickness they're using for the brass shim?
Yeah, use the use one or the thinner one. There's in the packet. There's one that's fairly thick. It still cuts, but let's go for things to be easier rather than harder, especially since this is the first time. So so the thinner shims would be better. Okay, for those that have color packets, okay, with a pencil on the back of it, do this. All right, let's see. Do that. And what we're doing is making some tracing paper. And then once you have that, that's it. Okay, just hold on one second. Can you get a piece of tape for this?
So far I've seen on the video, people are working with the brass shims, but we have several people that don't have video on. So if you're using the color swatches, can you let us know in the chat, and then we know how many people are using those?
Yeah, so I've got one of the color swatches here, and I'm putting it over that. And what we're going to do is we're going to trace it just like we traced the metal, but this will have a it would be like carbon paper. So, thank you. So once again, you're tracing it with the ballpoint pen. Now, did everybody who has color swatches, did they get the part about the using a pencil to draw on the back of this?
Yeah, so right behind that drawing, you took the pencil and just kind of scribbled over the whole area, and that's how you're making that transfer paper, essentially.
Ah, so there's our little anemone. See?
Oh, neat.
The next step. So what's nice about the copper or the bronze is see how whatever direction you go, you know, it reflects. Well, we can do a similar thing with the color swatches. So with your glue stick, glue the back. And glue the back here. And then put them together like so. Now, the best thing to do is to wait for these to dry, but seeing as we're running out of time, you can just jump right in.
An odd shape to cut out.
Yeah, it is. And I'm doing it quickly, and please take your time doing this. The next part is fairly simple. It's just we're going to make a little hook to hang our lovely little anemone and on.
Okay. It looks like folks are ready with their cutouts.
Everybody's ready? Cool.
Oh, they look like little baby anemones.
They do. And let's see. So you should have a good eraser. This isn't as good as I'd like, but then you can erase the pencil from the drawing that you did. Now, if it is all the same, I think the brass is a little better. But but the paint chips, you know, they can also work. Yeah.
It looks like just about everyone has brass for this class.
Okay, good, good. Yeah. Okay, and then, excuse me one moment. Okay, now puncture a little hole in our anemone. Ow, ow, ooh, ooh, ow, ow, don't do that. I always love that Far Side cartoon where there's a fisherman. No, there's a Martian catching a human by the mouth like we catch fish, and the thing says, "Don't worry, they don't feel anything."
Oh.
Yeah. Okay, and then.
And what are you using to poke the hole?
Oh, I'm sorry. I should have told you that. It's a push pin, which, as we now know, is good for toothpicks too.
It looks like. Christine, were you successful in using a toothpick to create your hole?
And then. And I'm just cutting a small piece, about like that. And I'm bending it one way. And then I'm bending it 45 degrees the other way.
Oh, John, you're just off of the camera.
Okay.
There we go.
And I'm bending it 45 degrees, so it looks something like this. And this actually isn't a very good hook, but I think for, since we've kind of run out of time, let's see if this will suffice. Yeah, see, this isn't going to work. I don't think it will. Let's see if it works. Well, I guess that doesn't look too bad. See?
So you made a little loop, and it looks like maybe there were 2 loops in that. And you hung the brass shim in and then attached it to the wire frame of the larger anemone.
Yeah.
Can you show the piece that you made to hang the shim again?
Do what?
Hold it up a little higher. Can you show just the little loops that you made to attach?
Let me, why don't I make one more? Just and here I am, I'm just like cutting the wire. Then I make one loop one way. And then at 45 degrees, I make the other one the other way. So they aren't on the same plane. So.
Oh, so it's like an S shape kind of, or it looks like if you twist paper clip, that kind of shape.
Yeah. Oh, hey, that's beautiful. Who is that? Stephanie. What's up? That looks great.
Yay. Oh, it's fun how that brass shim dangles in the middle.
So this Saturday and Sunday, if you want to work some more, I will be available. If you want to work on an enemy, that's fine. Or we can go to other things like the canary rockfish. That's a pretty cool creature.
And that PDF file has the other creatures in it, so you can look through there and see what's there. And certainly you can, if you're feeling confident, make some on your own, and or join John in making some more this weekend.
Yeah, I think.
And yeah.
I think you should call John and do a Zoom meeting with him, so he doesn't. That's what I think.
And I will note for those of you, there are several of you participating in all 5 of the events, and so we're making different creatures for each event. So there's a handful, about 4 to 5 for each event that we'll be making. So each of the art classes will be different.
So what we have is Bree was able to contact people and we got 5 creatures for each location, and we will do. So the 5 pages that you got, or 7 pages, I think it was the 5 creatures looking for something here, the list of creatures. Oh, here it is. So, so over these next weeks, we'll be doing the stellar sea lion, the black oyster catcher, marbled marlet, sand dollar, kelp greenling. Giant Pacific octopus. Now, how cool is that? Oh man.
That'll be a complex one to wrap.
Yeah, but boy, will it be worth it. Well, I'm glad it's one of the last ones, and that way you can get more complicated.
Yeah, I saw some excitement with that octopus, though.
Yeah. Oh boy, Cape Perpetua. That's like you got the stellar flat and the giant Pacific octopus.
All the complex ones.
Yeah. Well, everybody, I think that's it for today. What do you think?
Thank you so much for sticking with us through the education and the art class. Again, we'll be following up. Oh, here's our host is back from he was really concentrating on the art there. I'll hand it over to Charlie.
Oh no, thanks, Bree. Thanks for helping kind of guide us through that, through the art portion of the project. And thanks to everybody for participating. Thanks to John for guiding us through all of this. We're really excited. And again, if you guys have some feedback, keep it constructive, but we'd love your feedback. This is the first time we're doing anything like this. And so we're all kind of trying to figure it out. But it's been really awesome. And thanks, John, to you really for bringing this idea to us. We get boxed in sometimes in the world that we live in, and it's really been great to bring some art to the world of marine reserves and the work that we do for conservation. So thank you for your great collaboration on this event.
Well, I think it's very exciting that, to do 5 marine reserves and to learn about them and also do some art about them.
Awesome. Well, thanks everyone. That concludes our first Creatures event. We have 4 more to go. Hope to see some of you next Wednesday, and we'll be following up with more information. So thanks again for joining us.
Thank you.
Thank you all. Have a great night. Bye.
Bye.